All right. So I wanted to start off by introducing myself. Hi, everyone. I am Frances. So my background is in the life sciences. So I did my undergraduate at the University of Alberta studying physiology. I was really interested in how the body works and different situations in which it doesn't work. And that led me to here in Toronto, where I did my PhD in physiology. And that was really studying how a single cell grows into multiple cells and then makes us all these correct decisions in order to become a baby and the placenta. And we were really fortunate during that time because that was really the bloom of all these single cell technologies. Because I was working on for early first trimester placentas, and they're about this small. And we were really challenged by how could we study such minimal tissue and very rare tissue that we have in Canada. So by the end of my PhD, this is a cell atlas of about 45,000 single cells that we can follow through different weeks of gestation to see how they make these lineage decisions. Working with a lot of human data, seeing that we're getting a lot more genomic technologies in clinical and personal applications like, at-home genetic testing kits, and being on the data analysis side of it, that's what motivated me to do a master's in bioethics at the School of Public Health here as well afterwards, which was really interesting to kind of tease apart and spend some time thinking about what's the best way that we can move this technology forward in a positive light, as we've seen a lot of technology kind of like rush forward. And we want to make sure that this is a very valuable tool that is used correctly. So I've spent a couple years at the University of Toronto and Toronto Metropolitan University, specializing in getting some more computational classes started. So at UTM, so University of Toronto, Mississauga, we got first year classes of over 1,000 students all up and running in R, which was really amazing to see. We're replacing Excel with R and Python and getting them up and running with data science tools. And I've recently moved over to the Center Hospital at the University of Montreal as a bioinformatician, joint with Juniper Genomics, which is an IVF company working on advancing some genomic screening of early embryos to try and make more informed and educated decisions about which ones will have the best track developing forward. We also have our instructional team, so if you'd like to introduce yourself.
Yeah, so my name is Zoe, and I'm a PhD student here at OCR and at the University of Toronto. And I have been TAing this course for 3 years now. I also TA some bioinformatics undergrad courses at U of T. And my project is mostly focused on looking at RNA-seq data from cancer metastases and trying to figure out like either where the primary tumor was from the metastasis or vice versa. So if you have any specific questions you want to talk about your project, I saw we had a couple people doing cancer kind of stuff. Feel free to come up to me at lunch or whatever. I'm always happy to talk about my work or your work, and yeah, excited to be here.
Awesome. Thank you. And thanks for coming again.
Anytime.
Don't worry, I was in your shoes many, many moons ago. I didn't have any computational background in my undergrad, and I started this when I started grad school. And this was one of the first workshops that I took here many, many, many moons ago. And I did back to back the intro R and I think the RNA-seq one that they had at the time. And I know it's an intense week. We are going to just focus on the foundations here. And this will give you a good foundation and a platform for moving forward to the different techniques that you wanted to apply. But I want to give you the confidence to explore code, maybe edit some code. We're not going to shoot for the moon because this is learning a new language. So I know nothing about Spanish. If I was going to take a 2-day workshop on Spanish, I would not expect to be able to speak to somebody fluently. Or I think practically, I don't think I would be able to really make sentences, right? You pick up some words, you learn what are good resources, and have that confidence to keep learning. So kind of like navigate your expectations of what you're coming out of a 2-day workshop with. This is going to help you learn more, but you're not gonna be running RNA-seq flows at the end of it, but I want to move you in the right direction so that you can explore it yourself, either independently, looking at scripts online, or in further workshops that will blend from this one.
What about
Which one is? Yeah. La bibliothèque. Yeah. Now that's immortalized. Yeah. Okay, so This will mostly be a code along workshop, but I have a bit of an introduction for why we're all here. And we all have our own motivations, but we're here because we see the value in big data. Over the last, I'm going to say maybe about decade, we've had a big increase in data collection, storage, and analytical power. So if you think about even all the smartwatches that you're wearing or your phones that are measuring your steps, we're getting data constantly and it's being stored somewhere. And it's a wealth of information that we can analyze and make use of. But the more data we have, the more complex our analyses are going to be as well. We thought it was amazing to have the genome, and now we have it at a single cell resolution. That's all going to cause more complexity in how we're handling it. And it blends into that reproducibility crisis if we're all using different strategies. So we have a lot of challenges in big data because of the size and complexity of these research projects. And I'm sure you guys are probably here because you see some aspect of that that is challenging for your work. And I love kind of putting this into the context of the Human Genome Project because it first began and the idea was set in 1990, and it only took about 10 years for it to first be published. And then it was fully completed in 2003. So this was a project that cost billions of dollars. They took the data, recorded the data, and actually stored it in hard drives that had to be sent physically around the world because no single computer could analyze a genome in its totality. It was an international effort, came together. And even at this time, I think 2 to 5% of the budget was set aside for the social and ethical and the legal implications of sequencing a genome, knowing that this was really going to shake the landscape of how we are using health information. So after in 2003, when we had the complete mapping and understanding of all the genes, that opened doors to what we are looking at in terms of reference genomes. But if we think about today, you can literally, at least here at U of T, cross the street and have a facility sequence a genome in a couple hours for a couple $100s. Or go online and order one of those DNA testing kits that doesn't get us the same depth, but it's like on sale for $99 at Christmas. So the technology is moving so rapidly that it's increasingly affordable and we're getting more depth of information. So, and this was really only like 20 years ago that we had this technology incepted. So the tools are now moving forward to try and catch up with the complexity of this data. And you know that the genome is being used in all these different contexts and even above epigenomics, moving it to kind of complement data in the DNA and in the chromatin conformation. So the complexity of this data is only growing. In this workshop, we're going to be talking about R. So this is a little bit more on the technical side to make sure that we're on the same page. R is an open source programming language. So open source means it was developed and all the back end of it is open for you to see if you wish. Now, we don't normally peruse through everything that's going on. That's not really our job. We can let it be a mysterious black box for a bit. But if you're running a t-test, you might want to know what groups are being compared. So this is an advantage of R compared to something like MATLAB, which has its own proprietary functions. And sometimes you're not really able to check what it's doing underneath the hood or make some modifications if you want to do a slightly altered test. In R, everything is open to you if you wish to see or change. R was based on statistics, so it's particularly good if you're running a lot of statistical analyses. And they also have a high concentration of people that are interested in visualization. So I think that really complements each other. When, for example, if I'm making a projection of 45,000 single cells, the data can get confusing. We can't show everything all at once. So I really love R's visualization tools to be able to communicate that end. If you're the only one that understands your results, that's not really good enough, right? We need to put it into a paper, show it in a presentation, and R is great for that. R was started by Robert Gentleman and Ross Ihaka, and one of these guys actually was, I think, the CEO or an executive of 23andMe's bioinformatics side. So again, it kind of reflects that utility and the landscape that R is being used in.
That's why it's called R.
Yeah.
Start with R.
Yeah, so R started, I think, based on, I think they were kind of like reflecting C. So they kind of played on that name of like a single letter, but it was also because both of their names were R. Yeah. You'll see that because the R versions are also named, I think, foods. And some of them, I remember one year the version was like Christmas pudding from like Snoopy or something. So they're still quite lighthearted as a development group. Yeah. R, I know you might not believe me, but it's relatively simple compared to some of the different programming languages out there. The grammar rules are quite well defined, and compared to other languages, it's a little bit more human readable in how you write it out. Everything is object oriented. So we're going to take some values and store it in an object, and then we can multiply that object, put it into a new function. So we have these virtual objects that we are going to store and manipulate values in. As I mentioned before, highly customizable because you can see all the code and there's a really large user base. I've been teaching R for a very long time. I will guarantee you will not break R in a way that hasn't been broken before. So feel free to experiment. This is your first maybe like week of intensely getting into it. Don't be afraid of damaging anything. You can always read the file back in. If you run an error message, I guarantee someone has done that before. And I think sometimes new beginners also get frustrated because it's hard to find answers online. Learning how to like Google your problems and understanding the error messages is part of the journey along the way as well. So we'll go through some errors, we can walk through them together, and hopefully I can help you get more comfortable on that side as well. Now, the other thing that you should have installed is RStudio. So R is the language, RStudio is the environment. Do we have that? Okay.
I think you had us installed, right? Like
Yeah.
RGB.
R GUI should be just the language. Do you have RStudio?
No.
Okay.
I thought I ticked all the boxes. We can help you with that.
Yeah, we'll get
Don't
you
worry
caught
about
up.
it. Okay. But yeah.
So RStudio is like the editor in which we are going to be using R in. It has changed. I'm not sure if it officially changed names yet, but it used to be underneath this tidyverse. So tidy is a function in R. There's a bunch of related functions that they call tidyverse, like the multiverse. But to make a more neutral branding, they renamed it a few years into POSIT. So you might hear those names being referred to. RStudio, they wanted to move away from just R coding into POSIT because it can actually run R, Python, Terminal, Bash, many different languages inside. With the acknowledgement that such as Jupyter Notebook that you might have heard for that is more popular in Python but can be used in R, some of the analyses you'll need actually cross different languages. So it's really nice to have all your code in a pipeline at the same time. It also helps because there's something called syntax highlighting. So if you have objects and functions that act on it, they'll be colored differently, so it's easier for you to identify. And we'll show you some examples when you actually get into R. And we can manage multiple projects. So if I'm working on my own data and I have another project that I'm analyzing collaborators' data set, I don't really want conflicts between the workflow, and I don't want to have like multiple laptops going at the same time. So you can have different projects wrapped up in R. As I mentioned, you probably have heard of Jupyter Notebook for Python. This is another editor that can be used, but we'll be using RStudio. And if you don't have that, we'll get you up and running.
Is that for the classifier?
Yeah.
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 And just like RStudio, that's
You're on Windows? Yeah. Yeah. So if you just like click your Windows key and type RStudio, you should be able to see it. If not, we can double check.
yeah. Okay. I'll like.
Okay. So in the way we are using these two things together, if we have English, when we're learning English, we will need to write essays and we'll use Microsoft Word for that, right? Microsoft Word doesn't teach you English, but it's how we communicate in English or other languages. Today, we are learning R as a language and RStudio as the editor in which we type our code in. So at the end of the day, that skill that you're really learning is R, the language, but RStudio is needed so that we can talk and manipulate things within it. You don't need to be in RStudio. I mean, like for outside, it's much preferred for this workshop that we're all in RStudio. But you'll find that some tasks you will do baseline R or running R from terminal if you have really large data sets that are going to lag because RStudio has an extra layer of interpretation. So sometimes when I'm getting to really big files, I'll have to move to terminal. Or if you're moving or you're using a remote server, that more likely you'll have to move on to terminal. So here I have overviewed what you have signed up for workshop objectives of different skills in R that we're going to learn. But I wanted to also draw your attention to the bottom of some maybe soft skills that we are going to have to work through together as well. Getting used to typing with 100% accuracy. This isn't something that we do in day-to-day life, right? You can imagine that if you're like texting someone, sending an email, or even writing a thesis, you have time to go back and edit for grammar or clarity afterwards, but that draft of your manuscript will still get the idea across. When you're coding, you need 100% accuracy for that code to execute, or it'll give you an error message. And this is frustrating when we're not used to it, right? And very small changes like a dot, a period turned into a comma can be a little bit difficult to see. A misplaced capitalization also thrims, or most of the time. So these error messages are not indications of your failure as a coder. It's R's brute way of communicating with you. So anyone who codes, we're encountering error messages. R just doesn't really have a politer way of saying that to you. So don't let it get you down. It's not that you're not meant to code. Coding is just a bit frustrating getting started. And I think something that a lot of new beginners also struggle with is that like speed of coding. So I'll try and slow down a little bit. But if I am going too fast, just let me know and I'm happy to adjust with the flow. We're going to learn to interpret warning and error messages. R always says it in the most scary red font, but it is, again, a different language, so we need to learn how to understand what the computer is telling us. They're trying to give us hints to what it doesn't understand, but we need to learn to interpret it. And as I mentioned before, I hope that at the end of this, you feel comfortable and encouraged to continue exploring R and programming. We're not going to be able to probably finish the whole analysis on your data set at the end of this workshop, but I hope you're motivated to move towards that track and keep going with R. So we have these two stickies for everyone. What we're going to do while we're going is, as we're moving along, we'll be doing some code along. I'll give you some time to do exercises. If everything's okay, it'd be great if you could just have the little green sticky up over here. And especially for exercises, I might say everyone take down your flags. And when you're done the exercise, let me know that you're done with a green sticky, or let me know if you need help with a red. This kind of prevents you from having to like keep your arm up if you're stuck on an error. Zoe will be looking out for these flags as well. We're small enough that you can probably flag her over if you see that she's free. But if something's unclear, please let us know. We're going to try and move through the workflow together at the same pace, and I know we're going to have a little bit of differences, but if you have an error in a code chunk that you can't move past, and I'm moving on to the next one, that's your time to just flag it so that you can keep up, because everything relies on the previous code. So if you have any small errors, you've tried it a few times, like I would say maybe like 3 to 5 times, you've tried different edits and it hasn't been able to fix it, just let us know. And we can talk about it together because you're not the only one that has had these errors. I wanted to also just go back to this code of conduct before we get started with our learning material. I am asking you to do something that is, it sounds like outside of the scope of what you guys normally do, right? We're learning a new skill and everyone is welcome here regardless of your identity and your experience. So just be very mindful. We're giving feedback and communicating with each other because we want this to be a respectful and a safe space for exploring a new tool. Generally, this is a great environment, a very supportive. We have a lot of help here for you. So if you're ever feeling a little bit kind of like discouraged with the code or you need a little bit of help, we're here ready to help you and get back up running. Okay, do we have any quick questions before I move into a little bit more technical?
How, AI.
Mhm.
How do we use, a lot of people are now using AI and R together, and I'm just wondering if you're going to spend some time if we have time.
Yeah.
In the workshop to address this a little bit, or like what.
Sure. I'll maybe give like a high level response right now. I'm happy to chat with you maybe a little bit further in a break or something, or at the end of the workshop. Coding, I think complements AI a lot because there's a lot of repetitive tasks. And maybe this is great because I can also segue into everything that I'm showing you. I'm not asking you to memorize, right? So I'll be showing you a lot of different functions. You don't need to memorize them. You need to be aware that they exist so that you can go back and reference them later. For example, I'll show you how to make a scatter plot, a bar plot, a box plot. You just need to know that they exist and you can look up the code later to reference it. So I think AI is helpful for kind of like bringing up some reference materials for you. If you're saying, hey, can you show me like some references of how to make a box plot so they can apply it to yours? It saves you time from kind of blank Googling if you're not very used to finding that kind of answer online. I highly don't think that it's going to replace our jobs when you're in like a high level situation. I see it a lot in context for when I'm teaching like an undergrad class and people think that this is like their answer to finishing the course, but it's not. You're missing that middle learning part, right? You can kind of like skip to the end, maybe get an answer, but the moment that it throws you an error and you don't know how to code, you're stuck, right? Everything you still have to apply back into your data set. And especially my field working with human data, you can't rely, like you just can't integrate it that fully because your data is your own, right? Like there's a lot of protections over the data. So I think there's a lot of concerns in using AI for those protected data sets, which is something that I'm very cautious about. So I think it maybe in totality, it's a good reference material, it's a helper, but I don't think it's going to take over the world. Yeah. In this scope. Yeah. Okay. So R fundamentally is a calculator. So what we have here in the purple is the lines of code that I'm running, and the black is the output. So what I've circled here, these little arrows, we call them prompts. They indicate when R is ready to take information. So when you have these prompts, then you can write some code. For example, at the front, I've typed 2 + 2. You press enter, the code will run, and it'll give you back an output. You'll notice that it's numbered. So we have a square bracket with an I. It's saying you asked one question, your first answer is 4. So it always kind of like numbers it for you in case you were asking multiple things. So if you gave it a string of numbers and you asked to multiply all of them by 2, you would have more indexes that tell you the location. There are some pre-built values. So pi, and you press enter, it'll give you the value of pi, 3.141593. And we can also act. So this is what we talk about when we say R is an object-oriented language. The value of 3.14 is stored in the object called pi. So we can retrieve the value of pi by just putting in its name or acting on it by multiplying it by 2. R is very flexible in the spaces compared to a lot of different languages. So if you add a space between like pi space times 2, it'll still interpret it correctly. Other languages are not as flexible. The other key thing that we want to keep in mind is put arrow. So the syntax for reading this line of code is I'm going to put the value of 123 into this object called X. So when I type it and I'm typing along with you, I'll say X, put arrow, 1, 2, 3. Or sometimes people find you have to read backwards because you start with the object name and then you're shoving a value back into it. You'll notice also in contrast to over here where we say 2 + 2, it gives you a result back. When you put a value into an object, it doesn't have any black return underneath it because you didn't ask it for anything. You're saying, I want to store the value. If you want to double check that the value is in there, you need to type the name of the object and it'll give you back, now it has this value. So notice also here, we didn't predefine that X is an object. Before in the universe of that R environment, so it's a virtual environment that we have a bunch of objects in, before X didn't exist. we define X as an object by directly putting some value into it. So if in this environment I pressed A, enter, it would say object not found.
So you're saying something like pi would already be in there?
Correct,
There's some stuff that just is in there baked in.
correct.
But like, do we know intuitively, or is there a resource that like?
It would be an impossible list to make. So there are some defined values such as pi. There's also a function called mean that runs the mean of a bunch of numbers, right? So in general, don't save over names that could be something else, right? Yeah, but so I'd make things unique. You also, there is a function to run sine and cosine, right? So you would never want to save a value to an object called sine. Unfortunately, because it is open source, that means anyone can make anything and publish anything. So if you wanted to be a jerk, you can make a package called alphabet and be like A value 1, B value 2, right? So there always can be someone that makes a value of something. So it's an impossible list to make. This is something that you're just going to have to play around with, and it'll be very different for each other because you'll be using some functions that I don't use. So then you'll have different presets in your environment compared to what I'm normally working with.
Want to use that jerk down that alphabet.
Yeah.
You still have to download that package,
Correct.
right?
Yeah. No, it's not going to show up. Yeah. Yes, correct.
Oh, okay, so that's part of it too, is that like some stuff's baked in, but like most stuff's not baked in.
Yeah, so like you can think about,
I
yeah. Yeah. Yeah.
see.
You can think of R also, it's like a statistical tool at the base. So most of the things that are baked in there originally have to do with stats. So like the pi, the cosine, the mean function, summary, basic plotting stuff. But if you're running something that is like RNA-seq specific, so there's no predefined term for genome. There's no predefined term for maybe what's something that's very specific that we would use. I don't know, like single cell plot, right? There's no predefined term in base R for that. But if you read in a package about single cell sequencing, there might be a function like that. We'll go into how we get those functions. Exactly. Yeah. Yeah. There will be conflicts, and at the end of the day, if you do accidentally make an object called pi and overwrite that, you just restart the environment. You know better for next time. Yeah. Yeah. No, that's what I'm saying. There's no, you will not irreversibly break it unless you go in and like delete a source file or something. But in the environment of where we're working with, if you overwrite something, we can always get it back. Yeah. So the two fundamental things we're working with at the beginning are objects and functions. Functions act on objects. So objects have our values. Functions are just the tools that we use to act on it. So here, I'm making an object called prime numbers. So it doesn't have to just be single letters. When I'm coding for my projects, I maybe have too long of a name because I want it to be unique. So there's no pre-existing object of prime numbers. I'm taking the string of letters of prime numbers and making an object called prime numbers. We have our put arrow. So we're putting everything to the right of the arrow into the object to the left of the arrow. And I'm using a function called C. C means combine or concatenate. Sorry, side note, you don't need to type this because we'll be doing it together when we get into the workflow. I just wanted to talk through some of this first. So what it's saying is I want to combine all these numbers and put it into the object called prime numbers. So then afterwards, when I recall that object, it gives me all these different numbers. We can also run functions like class on the object. So I'm saying, what is the class of it? It's numeric because it's storing numbers. That means we can do math on it. This is in contrast to something that might be like tumor status, benign or malignant. Those are characters. We can't do math on that. And we can also run basic kind of baked in functions like mean. What is the mean of all of these numbers? Okay. One of the common mistakes that we have here are going to be those bracket pairs. So you'll notice that we know what is a function because it is always coming before the brackets. Functions are applied to whatever is in the brackets that follow it. If you don't complete the bracket pair, R will be left hanging, and that little arrow prompt will not start on the next line because it's waiting for you to finish that line of code. This goes back to some of the conversations we were having before. How do we know what functions are in there? So you can imagine on a phone, we have some applications, but there's always more. So we have an app store that we can pick the different tools or functions that we might need. You'll have to download it once that first time you want it, but every time you want to actually use that program, you still have to open it so that it's active on your front page, right? So you can download more apps than you're going to use in your day-to-day. That is similar to R. So we have a whole repertoire of packages. Again, anyone can develop these. Where you downloaded R from CRAN, the Comprehensive R Archive Network, this houses a lot of general functions. Different plotting functions, general tools, some analysis, some more specialized stats. If you're doing more focused biological applications, so I know in our lab we had a biorad machine that ran qPCR that they generally could only get analysis on the proprietary biorad software. Somebody has made an open source version of that. And since it's very biology focused, it's in Bioconductor. So you can think of it maybe like the Toronto library and the Vancouver library, right? Different ways and like different houses for a bunch of different tools. So if you find one that you like, you can identify where its home is. You'll have to download it the first time. So today we'll be downloading some packages, but you'll have a copy on your laptop. When you need to use it, you still need to open it every time you open that RStudio environment. So that's how you can control. If I'm working on RNA-seq data today, I'll have only the RNA-seq packages open. If I'm doing some visualization the next day, you'll just have a different set of tools. When you're starting out, maybe at least in like the first year or two, generally reading in more functions than you need is not an issue. Better to have them in case you need them. And it's better to read them in at the start because it kind of sets up your tool base for your day of working. But you might find as you do very complicated analyses that you have too much in your environment and you'll start to have to like narrow down what you actually need. Okay, last slide before we actually get into RStudio. If you have RStudio in, you'll see it's something like this. There'll be 4 windows. If yours aren't in the same order as mine, don't worry. It's something that you can kind of play around with and get into kind of what you personally prefer. The main window that you are looking at that is R itself is a console, so the top right corner over here. This is where I've been showing you those screen captures of prompt, response, prompt, response. All the code that you're running is calculated in the console. But if you think about maybe if you're a statistician that only uses a calculator, for example, and you're running all of your analyses on that calculator, you're going to forget what you did 20 presses ago. But you want that record so that you can go back and say, like, this is how I analyze the data. So on the left here, we have the script or the notebook. This is where you write it down. So in general, we write down our code in the script, and then we push that code to the console for it to be run. But the script is our living document that records what we have done. It might seem a little bit redundant at the start, but as you are figuring out your code, you might run a line 5 times to edit it and get it to do exactly what you want to do. You don't want to save like the 5 failed codes before you get the 6th one, right? So the script, we're just saving what works in a linear fashion. The console is just going to be buzzing in the back as like a permanent record, well, semi-permanent record of everything you've done. So those are the 2 windows we're going to be looking at. The environment is also helpful because it has a list of everything in that virtual environment. So all the objects you've made. If you forgot the name of an object, you can go back and refer to it here. And then in the bottom right, we have the help. This is also going to be where we get some of our plots coming up later when we do that. But we're going to be working with a markdown document, and it's really nice that it integrates our code and our results in the same window. that minimizes the need to have many of these plots open.